9 PhD students and numerous undergraduate students, and he's published 76 peer-reviewed papers, including in highly impactful journals like PNAS. So, Dr. Begley,
thank you very much for the kind introduction. That was really wonderful. It's great to be here. Louisville's a beautiful town. I've been well fed, had great hospitality. I had a little bit of, I think that was rye, right, Chris? But it's been a lot of fun. I'm a pretty casual guy. Call me Tom. Please stop me during the talk. Love to answer questions and learn more about what you're all doing. I'm going to tell you a little bit about research that's been ongoing in my lab probably since 2004. This is the research I decided to do when I became an independent investigator. My background really is in DNA damage and DNA repair, and I joke that it's my first love, and I'm working on RNA modifications, and I guess DNA is just an elegant RNA modification at this point. But I'll talk about stressing out with the Epitranscriptome, implications for exposure biology and human disease, and nuts and bolts. I'm going to be talking about how RNA modifications are dynamically regulated and how we think at the tRNA level, which is primarily what we study, how these modifications are used to regulate translation. And we get into the nuts and bolts of translation looking at codon bias. And this is the collection of codons in an mRNA sequence that specifies specific amino acids. With the understanding that codons are degenerate. There's many different versions of specific codons for specific amino acids, and there's interesting combinations that can be regulatory. We do this in a number of different systems. We've got yeast, bacteria, mouse, human cell line, and tumor studies that we've done. And I'll show you a couple vignettes of studies from really from yeast, mice, and humans that we've been working on the last couple of years to try and get some mechanistic information on how specific RNA modifications regulate translation during stress. And stress can come in many different forms. Before I go on, let me just it's not moving. It just worked. Well, while we're getting that working, I can give my disclosures. So I do have some commercial interests. We've Hovana and Codamax, which you'll see on the next slide. So Codamax is pretty new. We're really interested in how we can harness the epitranscriptome and codon usage to improve the production of biologics. I can say Codamax has a lab operating out of Worcester, Massachusetts. Okay. Hovana is a small molecule company that's interested in reprogramming tRNA to fix different disease states with a specific emphasis on cancer. And I do have interest in both. What I also want to do is I want to acknowledge the people that have done all the work. I basically sit in front of the computer and ask people for money and fill out reports that I think many of the established investigators can commiserate with. I've been really blessed to work with a number of wonderful students and postdocs through the years. They're pictured here. Sorry about that. Pictured here. This is the current group that I'm working with, really talented students from around the country and from the world. Also have to acknowledge the funding agencies that support the work. A big shout out to EHS, which gave me my first grant back in 2006 and have been a real champion of RNA modification work and epitranscriptomics. I've had the benefit of collaborating with a lot of established investigators. Big shout out to Jinjin Ding, who's now at the University of Arizona, who we've done some exciting work with. Also got to acknowledge Ph.D. from MIT. He's my longtime collaborator on this field. Many of you know him as a world expert in DNA addict analysis. I helped convert him to RNA modification analysis back in 2007, and he's a wonderful collaborator. One of the smartest and most generous scientists I've ever worked with. So if you ever get the chance to meet Pete, He's super friendly and a great collaborator and a great person to work with. And also for people looking for postdocs, he's an amazing postdoctoral mentor. Let me tell you a little bit about how we got into this field. So my background is really in DNA damage and repair, and we started studying cellular responses to different types of DNA damaging agents. We know DNA damage and stress is linked to lots of different human diseases, and the types of damage that we've studied over the years, can be ionizing radiation damage, ultraviolet light, different types of alkylating agents or compounds and toxicants found in cigarette smoke, ionizing radiation from different types of medical procedures, and also different types of pharmacological stresses and toxicants that arise when you have overdoses. At the very root, a lot of these can cause DNA damage and changes in reactive oxygen species, and these changes in reactive oxygen species can damage lots of different cellular macromolecules and disrupt cellular physiology. We study the regulation of gene expression in response to stress, and we know that essentially the blueprint for gene expression is built in DNA. The central dogma of molecular biology, I'm sure everyone's pretty familiar with it. And here it's, DNA to RNA to protein. It explains how genes are coded and turned into mRNA and into proteins, okay? We have our standard, what we say is our 5 nucleosides or bases here, the A's, the G's, the C's, the T's, and the U's that we're all very familiar with. We study the Epitranscriptome, which is really a regulatory system. It will control the when and how much of translation, okay? Many of you are familiar with epigenetic marks like 5-methylcytosine and histone modifications. These can control the when and how much of transcription. We're interested in RNA modifications that regulate translation, okay? Some example modifications that you'd find are Y, M5U, ribose methylated, Gs, M1A, M6A, and M5Cs. These are all pretty common RNA modifications. Lots of exciting work going on at M6A. I know here in Louisville, also at other places, this has been a really hotly studied modification controlling gene expression at many different levels. So, one of the exciting things about the epitranscriptome, it's really just a collection of RNA modifications. If you look across phylogeny and all species of life, there's 170 distinct modified nucleosides out there, okay? Any one organism probably has about 40 or 50 that can be found on the RNA. And this can be found on messenger RNA, ribosomal RNA, and also tRNA, shown here, okay? We study tRNA because it can be closely associated with translation, and the enzymes that modify tRNA are commonly called writer enzymes. There's usually writers, readers, and erasers. Writers will put a mark onto an RNA modification, an eraser will remove it, and a reader will bind to it and elicit some sort of signal, okay? When we talk about writers, at its simplest form, what we're looking at is a nucleoside that's written with a methyl group here, okay? And usually the methyl donor is S-adenosylmethionine or SAM. And the RNA modifying enzymes can be trins, tRNA modifying. They can be metals, which modify lots of different types of RNAs, and also ALK-Bs, which have a horrible name, but they can modify RNA also. Okay. So we're really interested in tRNA modifications, and tRNA is about a 70 to 80 nucleoside long RNA species. It has a classic cloverleaf structure. Importantly, it binds it binds with mRNA in the ribosome to facilitate translation. In general, each tRNA has about 8 to 12 modifications per tRNA. This is for your standard tRNAs. They're found throughout the tRNA in the acceptor stems, the variable loops, the D loops. We're really interested in the anti-codon loop, okay? So in general, tRNA modifications can regulate tRNA stability, tRNA structure. And when you start talking about the anti-codon and anti-codon loop, you start thinking about translational fidelity and translational rate. So translational fidelity is the ability to accurately decode a codon. So infidelity would be inserting an amino acid in the wrong codon, so it's an error. And then there's also rate, so that's speed. And when we talk about translational regulation, we're really thinking about fidelity and rate and the balance. We study, modifications found in the anti-codon and also position 37, because they have the ability to directly influence binding with mRNA, specifically codons and mRNA. And some of the modifications that we're looking at are these wobble U modifications like MCM5U. We also look at, we see as a hypermodified Q, or hypermodified G, which is the Queuosine modification, along with some others. But you see there's a lot of chemical diversity in tRNA, which offers exciting possibilities for biological regulation. Okay. First got into the field of studying RNA modifications and stress responses with some work we've done in budding yeast. This is old stuff, but the general idea was we were doing high throughput screens of yeast gene deletion strains to identify those that are sensitive to different types of DNA damaging agents. We kept pulling out yeast strains that were deficient in RNA modifying enzymes as being sensitive to classic DNA damaging agents. And the question was why, okay? We did follow-up work on this enzyme called TRIM9, which generates this modified wobble-uridine found on two specific tRNAs for either arginine or glutamic acid. And these tRNAs decode specific codons. And what we were able to show over the course of many publications is that these modifications are increased in response to DNA damage and other physiological conditions. And they're really important for decoding AGA and GA codons for arginine and glutamic acid. And if they're not there, there's some very specific transcripts that don't get translated very well. And they turn out to be DNA damage response proteins, some metabolic proteins and other, okay? So this started us on the path of thinking about tRNA modifications as regulatory units for different types of exposure responses and also physiological responses. This is where Ph.D. comes in. He's pictured here. Together with Pete, we worked out a methodology to essentially purify RNA from either untreated cells or tissues and treated cells and tissues. We purify the RNA. We do some QC and then some enzymatic degradation to nucleosides. We do HPLC and mass spectrometry, and it's quantitative. And we can compare the levels of 30 to 50 modifications from a treated to an untreated state, okay? The way we do that is we visualize things on a heat map, which is shown here. So essentially we have 4 different exposures of yeast here. And what we've got is modifications on the y-axis. And what you see is that the modification, if it's red, it means the modification went up in the treated relative to untreated. If it's green, it went down. If it's black, there's really no change. What we were able to show in yeast was that there was coordinated changes in RNA modifications that were specific to each of the toxicants, okay? And this was very reminiscent of what you would see with transcriptional responses to just toxicants. There was a very well-defined increase or decrease in specific modifications that highly suggested that this is a systematic response, okay, and it was a coordinated regulatory response. And we've done a lot of work in yeast to show that. I can say that we've taken this technology really to anywhere you can find RNA, and we've published studies in bacteria. This is a M. bovis hypoxia model showing changes in response in RNA modifications in response to hypoxia. We've done it in budding yeast to show that the response to different types of treatments that arrest cells in S phase, G2 or G1, give specific reprogramming of RNA modifications. We've done it in samples from rat livers from the NTP project. We've seen very specific changes in RNA modifications in rat livers. And we've also done it in HepG2 cells with different types of toxicants representing alkylating, oxidizing, and sodium arsenate's on there too as an oxidizing agent. So we see very specific responses in each of these species. We've extended it to other species also. This is an ingrained part of gene regulation. Okay. Now, I like to joke around at this point and say if you go back and think about learning about tRNA, we usually do it in biochemistry. As an undergraduate, you probably get about 20 minutes on it. Okay, and you rarely have told that it's so modified, but it's the most heavily modified nucleoside, the modified RNA in the cell. Okay, so. Now, when we start thinking about tRNA, we start thinking about, and especially modifications in the anticodon, we think about translation and codons. So many of us, or most of us, are familiar with the genetic code. We have 64 codons. 62 of these codons specify 21 amino acids. Good example would be, let's talk about lysine right here. Lysine has 2 codons, AA and AAG. Each of these specify the amino acid lysine and an mRNA. This code is considered degenerate because there's, for lysine, there's 2 different codons that specify the same amino acid. Now, interestingly enough, there's some amino acids that have 6 codons. So arginine has 6 different codons. So there's lots of varieties that genes can use for decoding purposes. And we started asking the question, why are there extra codons and why are there special codons? So a good example of a special codon is the one for selenocysteine. Selenocysteine is a 21st amino acid, and you'll hear about it a little bit later, but it doesn't have a specific codon in the genetic code, but what it does is it's a really elegant system for taking this UGA stop codon and recoding it, okay? And it requires an RNA modification to do that, okay? Now, if you start thinking about we'll call them the normal codons like glycine and arginine, classically there's been something called the optimal codon. This has usually been defined based on some classical and historical data as the most frequently observed codon. Many ways it's been considered the best codon to use for translation, but our studies and many others don't agree with that. Codon usage and codon optimality changes as a function of stress and a function of RNA modifications, okay? And what we've been able to describe in the literature is what we call as alternative genetic information and synonymous codons. So there's really a code within a code, and there's families of stress response genes that possess unique codon biases. So we'll find some families that have only using all of the A version of the lysine and the A version of the glutamine. And other gene families are only using the G version. So we've been able to stratify gene families this way and come up with a system, and it's really a regulatory system in which codon bias and RNA modifications are regulatory features for translation, okay? To do this, we've worked extensively with the Codon lab and a whole bunch of different technology platforms that include RNA modification analysis by LCMS. We've got advanced codon analytics to look at gene sequences across genomes and compare them. With a nice RNA sequence and informatics pipeline for identifying and quantifying small RNAs, which is quite complicated. And we also have some proteomics approaches for looking at translational regulation. And the general model that we've been able to build is that in response to stress, tRNAs are reprogrammed by these writers. Here we have it at the wobble position. What this does is enhance the translation of mRNAs from we shall say codon-enriched genes, and these are usually survival genes, stress response genes, metabolic genes. And what this does is promote sort of a just-in-time stress response. These RNA modifications are allowing cells to better use mRNAs for translations, and these mRNAs have very distinct codon biases. I like to make the analogy here that if anyone who's studying transcription factors knows that a transcription factor will have a preference for certain binding sites that have specific sequences. And that drives transcription of specific genes. Our model is analogous to that. We were saying that specific codon biases in genes are better translated in response to stresses, and that matches up with RNA modifications. That's the big picture of it. I'm going to take you some details of it from some very specific stories that we have ongoing in the lab. Okay. Big picture outline is here, showing you how the tRNA epitranscriptome is dynamically programmed in response to different exposures. Talk a lot about stop codon recoding and selenoprotein synthesis, and how it's really I think the simplest form of what I call codon bias translational regulation. Highlight some of the diet and microbiome links to the epitranscriptome, specifically with the wobble Q. This is a hypermodified G modification found in tRNA, and how it's really important for regulating ROS stress, mitochondrial physiology, and translation. We've done some really neat studies on looking at the most biased human genes, and it turns out that oncogenes fit this category quite well. And there's some really strong links to RNA modifications, codon bias translation to the translation of oncogenes, and also RNA modifications and writers being essential components of oncogenic programs, specifically at the level of translation. And I'll show you a neat paper by Richard Gregory on that. So there's some strong links to cancer etiology that we're exploring also. All right, so here's just a little bit on stop codon recoding and selenium. So selenoproteins are pretty interesting from my perspective. There's about 25 of them in humans and mice. They're found across phylogeny. In humans and mice, these selenoproteins have very distinct roles in redox signaling and antioxidant capacity. There's about 12 of them. They're the glutathione peroxidases, the thyrodoxine reductases that remove hydrogen peroxide and lipid peroxidation products. They're very important for managing ROS inside the cell and also mitochondrial function. Now, selenocysteine is a really interesting amino acid. There's not a specific codon for it. It is decoded in a really elegant pathway, but at its root, it's using this UGA stop codon and it's recoding it, okay? And to do this recoding, there's specific RNA modifications, and these are wobble U. This is the MCM5U, this is MCM5UM, okay? This is a specific wobble U modification found in tRNA for selenocysteine, okay? We built a mouse deficient in ALK-BH8, which is important for generating both of these modifications. Okay. All right, so just a little bit more about stop codon recoding. So to incorporate selenocysteine into mRNA, you need a very specific tRNA. It's tRNA that's charged with selenocysteine. This is a unique tRNA that has a really big variable loop, and it only has 4 modifications in it, as opposed to the 8 to 12 usually found in most other tRNAs, okay? I highlight what I think is the most important one, but I'm biased. This is the wobble U modification that we think a lot about. It is catalyzed by ALK-BH8 and accessory protein TRIM112 to make MCM5U, and then it's ribose methylated, most likely by a SNO RNA to get the ribose methylation. Now, stop codon recoding is really interesting because you have an internal stop codon, UGA, also an end codon. So it's codon bias, and that it has twice as many stop codons as you'd expect. It requires a specialized tRNA that has a modification. There's also the CIEHS element. This is a 3' UTR that helps bring translational machinery. So it's a really specialized form of translation. And it makes really important proteins for, I think for environmental exposures and other physiological processes that these are detoxification enzymes, okay? The slide that you're not seeing the format is here, but this is sort of the introduction a little bit to some of the codon analytics that we do. So we have the ability to download whole genome sequences, interrogate all the genes and count the codons in each, and then generate what we say is frequency measures. This is what's the frequency of using that codon relative to the other synonymous codons in that gene. That's anywhere a number from 0 to 1. You can get statistical values, and there's Z-scores. So positive is overused, negative is underused. And then we can start analyzing groups of genes and saying, well, what's the trend in this group? We get a t-statistic on a trend. So if it's positive, it means that group is overusing a codon. If it's negative, that group is underusing a codon. And if you look at the 25 selenoproteins and look for statistical enrichment of codons in that group, you get your UGA stop codon, but you also get a whole bunch of other ones that are primarily C or G ending codons. Okay, so there's a very distinct codon usage pattern. We can colorize that on a heat map here. I have all the selenoproteins up here. You probably can't see. Here's all the codons. What you can see is this group of codons is heavily enriched. Okay, and these are your G and C, some of your G and C ending codons and UGA. So we can say there's a really distinct codon bias in the mRNAs for selenoproteins. Now, we postulated that this codon bias is going to be needed for efficient translation of selenoproteins, and that translation is going to be dependent on a specific RNA modification catalyzed by ALK-BH8. We used a gene trap model to make an ALK-BH deficient mouse. We generated MEFs and tested these MEFs for sensitivity to ROS, and as expected, they were sensitive to hydrogen peroxide. You expect that if they're not making glutathione peroxidases and the thyrodoxine reductases efficiently at the level of protein synthesis, there should be this sensitivity. We then went and also looked at RNA modification, namely this MCM5U, in response to hydrogen peroxide over a 24-hour time course. We saw a significant increase in wild type and that's not matched by the ALK-BH8 deficiency. So further characterization of this, these maps, we're looking at translational regulation. Translational regulation is a real pain to analyze. You got to look at mRNA, you got to look at protein, okay? And optimally, you're also looking at the polysome, the actively translated mRNA, okay? I'm showing you the simplified version for, this is for GPX1. This is a selenoprotein. We're using qPCR here to look at the levels in wild type and the ALK-BH8 deficiencies. Very similar in both cell types. We go look at GPX1 levels under untreated condition. We see a difference. This is a immunoblot. We see a significant decrease in the ALK-BH deficient maps. If we look at the wild type response to hydrogen peroxide, we're seeing a significant increase in response to hydrogen peroxide that's not matched in the ALK-BH deficient model. Okay, so this is classically what you'll see for translationally regulated protein, especially the stress responses. There's sometimes a basal defect, but there's always a stress-induced defect. You see this for GPX3 also, and GPX6. So we've gone through, and there's a lot of the selenoprotein levels are disrupted in the ALK-BH8 deficient mass. But I will say, I'm not showing it now, we've also looked at livers and lungs, and it's tissue specific. Okay, there's tissue specific differences, and that's probably going to be based on mRNA total levels also. Okay. The ALK-BH8 deficient model that we're using is a little bit leaky because it's a gene trap. So I had a student, Sarah Epke, who came into the lab and was really interested in liver and heart and said, I really want to look at some of the organ systems in the mice we have. And my good friend Von Roos and I was talking to him in a meeting, and I was just probing him for what I should be doing. He's like, you got to use acetaminophen. This is the classic toxicant. You got to do it. I was like, all right, so I suggested to my student, and essentially we all know that a high dose of acetaminophen can be toxic. It can essentially generate reactive oxygen species inside the cell or organ. So what we did was we did a single a high dose exposure of our mice to this IP injection of acetaminophen of 600 mg per kg. And 6 hours later, we looked at markers of liver damage, oxidative stress, and also protein levels. What we can see is that in response to acetaminophen, this dose, we see an increase in all levels in the wild type. It's further increased in the deficient mice. So we are getting a liver stress. We see a trending increase in oxidative stress. It's not statistically significant, but most importantly, what we did is when we looked at the protein levels, and this is using the WEST system, which is a capillary electrophoresis system. It's really much better way to do immunoblots. I can totally guarantee that. And what we see is in the wild type, we see a significant increase in our GPX3 levels in response to acetaminophen, and this is not matched in the mice. Okay, and our ALK-BH8 deficient mice. So big question that we wanted to ask of that is, well, all right, so a lot is known about acetaminophen, and we're certainly not experts in it, okay? I'm definitely more expert in taking it for myself and administering it to mice, but we want to ask a really simple question. What's going on with RNA modifications in the livers, okay? And we were somewhat astounded with what we saw. Okay, because we saw significant reprogramming of RNA modifications in livers. And let me take you through this. So this is this heat map is all relative to wild type untreated livers. And what we see is that in response to acetaminophen, we see an increase in the wild type livers, a number of RNA modifications. Okay, our ALK-BH8 deficient mice under we'll say saline conditions, we see some changes in RNA modifications relative to wild type, but the response at the epitranscriptomic level is essentially the polar opposite. We see decreases in all these RNA modifications, which likely has to do with some sort of tRNA degradation. But what we want to point out here is that if we look at tRNA for selenocysteine, and this is from the global pool, we know tRNA for selenocysteine has 4 RNA modifications. It is MCM5U, which is my favorite here. There's I6A, which is in the anticodon, M1A, MY. MCM5U modifications are significantly down. We also see the same thing for I6A, M1A, and essentially also Y. So all 4 modification levels are down in the tRNA for selenocysteine, okay? So there's likely a tRNA stress going on in the absence of this modification. And this is affecting, I'll show you here, this is affecting the protein levels for a whole bunch of different selenoproteins, okay? We see GPX3, which I've shown you already. GPX1 levels are way down, and the acetaminophen treated ALK-BH8 deficient mice. Similarly, the thyrodoxine reductase 2 levels are down. Okay, so it's an interesting connection that we have now between RNA modifications and a response to acetaminophen stress in the liver. Now, I'm not going to show you the data on this, but we've done similar studies in the lung with naphthalene, so that's mothballs. You see lung-specific deficiencies in ALK-BH8 and wobble-uridine modifications also affecting response to naphthalene. And we've got some data on different types of cigarettes, smoking toxicants, and I think I show that next. But you know, the importance of selenoproteins in different types of responses is very well known. It's been in the literature. Okay, I think the thing I highlight here is we're connecting this now back to an RNA modification, one that's dynamically regulated as part of translational programs. And in my view, selenoprotein synthesis is the simplest translation program to study. You have a limited set of RNA modifications, you have a limited set of mRNAs, and you can really isolate one codon, UGA. But we know it's a lot more complex because there's lots of codons and lots of diversity in the codon usage in mRNA sequences. Oh yeah, so this is the data. So this is an ongoing study with Pete Didon and Jinjin Ding at the University of Arizona. But what we've done is we've taken our meth models and we've treated them with LD20 or LD50 doses of formaldehyde, sodium arsenite, cadmium chloride, and naphthalene, and just look for changes in RNA modifications. Don't have to take too much away from this, but notice that there's some big changes in response to cadmium chloride. Some make sense of that because transcriptionally there's not much going on, and it's different really for each of the different toxicants. Okay. Again, I think in retrospect, anyone who's done significant transcriptional profiling in response to different toxicants, this should not be unexpected. This is just the other end of gene regulation. But I think what it does highlight from us is, well, translational regulation is complicated, But gene regulation is super complicated. We're going from transcriptional regulation to translational regulation, and there's lots of different factors that can influence that. All right, so any questions on that before I sort of shift gears? Happy to answer anything that you have, but I'll tell you a little bit about the human epitranscriptome being reprogrammed in response to different types of exposures. And this is largely what I showed in SOT, but what we did with Hep G2 cells is we treated them with a host of different toxicants to look for changes in RNA modifications in response to each of them. And what we found is for all the toxicants that we could associate with ROS, we saw an increase in this one modification called Q. This is Queuosine. We zoomed in sodium arsenite here, and what I can show you is that this is an LD20 dose. At 6 hours and 24 hours, we see increases in Q. We also see T6A. This is a position 37. But this Q response was pretty consistent, okay? And we've seen it in other systems. This is an LD7. This is a heavy-duty dose, but where you see a time course where we're doing exposure in our media, then after 2 hours, we're switching the media and following it again, but you see this change in Q that's consistent, okay? Now, I'm a mechanistic biologist at heart, so I wanted to ask, well, what's going on with Q? Why is it as important? Now, Q is a really interesting RNA modification, okay? It is inserted into tRNA, and it's really 4 tRNAs that it's inserted into for 4 specific amino acids. The Q base is incorporated in what we call a transglycosylase reaction. So the Q base is swapped in for G by this QTRT complex. This is made of QTRT1 and QTRT2, and it's done in a very specific tRNAs, what we call the gun anticodon. Now, this Q base is interesting because mammals don't make it. All right, the Q base is derived from our diet. Turns out it's in a lot of sour cream and yogurt products, and also from the microbiome. Okay, and this is one of 2 examples of a tRNA modification that the corresponding base is supplied by the environment. Okay, so this is a diet microbiome associated epitranscriptomic mark. Okay. Now, interesting to think about how people's diet and microbiome can now affect RNA modifications. And I think that's going to be an important area. The other modification that's similar is, have you guys ever heard of taurine? So I asked my students, hey, where do you find taurine? They're like, oh, it's in my energy drink. So that's one is actually a mitochondrial RNA modification. It's very specific. So, but just go back to Queuosine. So we're really interested in Queuosine. And what we ended up doing is you can actually deplete Q base from media. Okay, it's found in serum. And essentially what you do is you dialyze serum. Okay, and you get rid of the Q base. And you do this, over the course of a couple of weeks. And essentially what we did is we dialyze the serum, and then we can actually buy the Q base from Sigma. It's really expensive, but you can add it back, okay? So we have our normal growth medium, which has sort of normal Q in it. We have dialyzed FBS, which is a low Q, we'll say. And then we have a dialyzed FBS plus Q medium. We grow our cells in that, okay? We purify the tRNA. We analyze for Q levels in this small RNA. And what you find is, this is the normal level here. Our low Q dialyzed FBS, we see about 100-fold decrease in Queuosine and tRNA. When we add Q back, we see about a 4-fold increase, okay? So here we started analyzing the cells that are associated with this. And when we did our viability assays, we saw that the low Q cells had a significant sensitivity of sodium arsenite, okay? And we see it for other oxidizing agents too. So to further, and when you add Q back, you can rescue, okay? So what we then did was actually start looking at ROS levels and cells grown in these media. We sort of have our normal, low, and greater than normal Q levels. Using Cellrox Orange, we're measuring ROS, and what you can see is that in the low Q grown cell, the cells grown in media that supports low Q and tRNA, you see a significant increase in orange signal, and this is more ROS, okay? Again, it's rescued back by adding Q into the media when it gets into tRNA. We then looked at mitochondrial mass using MitoTracker Green, and we saw significant decreases in mitochondrial mass. So there's deficiencies in the mitochondria in the absence of, or in the decreased Q cells, okay? Now, everyone, just to recap mitochondria is a really interesting organelle. It supports its own translation, okay? And Q-modified tRNA is important for translation in the mitochondria, okay? So, next thing that we started doing was we did polysome profiles, and these are really complicated assays where you're essentially fractioning the polysome in a gradient, and you run it out on basically in a chromatography system. We've done this for a lot of different conditions, and I'm not gonna bore you with all of it, but when we normalize our cells to the corresponding treated versions, we have our normal Q levels here, okay? When we add, when we have low Q, we see a significant decrease in global translation in response to stress, okay? And we see really more translation. All right, this doesn't work. There we go. More translation when we add, when we do the Q add back, okay? So this is telling us that really taking Q out of tRNA can have this real massive effect on translation, okay? And we wanted to look for something a little bit more subtle and mechanistic, so we went back to our normal growth media conditions, and we did a treatment with sodium arsenate. But what you find is that, again, we can highlight that when you do the treatment, you see a decrease in mitochondrial mass. Excuse me. And then what we ended up doing was a CIEHS analysis to look at really, the metabolism. So these are complicated analyses that measure mitochondrial respiration and extracellular acidification, but they can tell you a lot about the metabolic state of the cells. Is it in glycolysis or is it in aerobic metabolism? And what you find, and classically you see this in the literature, is that treatment with sodium arsenate poisons the mitochondria, and you see a shift to glycolysis to promote energy production. We followed these studies up with some proteomic analysis to look for proteins that are upregulated in response to sodium arsenate treatment in our HepG2 cells. Got about 80 significant proteins that are upregulated. Their classes are found here, largely metabolic of what we picked up. We find 7 proteins that are upregulated in response to sodium arsenate, all involved in glycolysis. They're located here. And it turns out when we do some sophisticated codon analytics, what we see is they're all biased for specific codons that end in C. Now, Q is important for decoding codons for 4 amino acids: tyrosine, histidine, asparagine, aspartic acid. They all end in a U or a C, they're codons. Okay, there's 2 options. All these proteins have the the C option. Okay, and we think there's a model really where what Q is doing is that it's an increase in tRNA in response to sodium arsenate stress and other stresses to promote the translation of transcripts that have these C-ending codons here, okay? Now, to follow this up a little more subtly and actually start looking at specific genes, we decided to ask some really targeted questions on codon usage, okay? And that's the transition here. So really what we wanted to ask is, what genes in humans have a lot of codons that either end in the C or the U for that are decoded by Q, okay? What we're looking here is what are the most biased genes based on codon usage in the human genome? Okay, what are the extremes? Okay, so this is an example of, this dates me a little bit. This is Where's Waldo? What you're doing is you're trying to find this needle in the haystack. What's the most biased 50 genes out of 30,000? Okay, I'm not going to go through the math, but what we can do is the top 2% of the genes based on codon usage, or we get the value. It's called a sum z-score. But essentially, what we did is we identified the most biased genes in the human genome based on codon usage. They're over here. Here's the codons. And essentially, what you see is there's two groups. There's one group that's massively overusing codons that end in G and C. There's another group that's overusing codons in U and A. But this bias is more extreme. Okay, so these guys start going through this list, which is interesting. I just want to highlight there's this one protein, one mRNA for a protein called CEDPB. Okay, if you compare the codon usage in humans of CEDPB to mice, you see this extreme codon bias is maintained. And we are really excited by this because what we see is that the codon bias really is found in these Q decoded codons. It's really overusing the C ending codons, almost totally committed to it. So you have, so for the CEDPB, it's a transcription factor for immune and inflammatory responses. It has 41 C ending codons, okay, for these 4 amino acids. Okay, that's a pretty extreme number. You think about the possibilities natures could use to actually code this mRNA. The numbers are astronomical. Okay, the amount of diversity. There's a group of different combinations of codons that can be used to make the CEDPB. Okay, why is one codon combination used, and why is it so extreme? Okay, that's the question we wanted to ask. Essentially, what we do is we do a classic codon engineering approach. So we've taken the CEDP mRNA sequence and call it the wild type is what we call the optimized sequence. It has the 41 C ending codons that are decoded by Q, and we swap them all to the U ending versions. Okay, we measure RNA and protein, and what I'm showing you is the protein levels here. And what you can see is when you change the codon usage, you see significant decrease in protein levels. Okay, so here this is a really good example showing that these C ending codons decoded by Q are regulatory. Okay, there's lots more work to do here, but what we're trying to figure out is what's really this code of codons in gene sequences. What are the regulatory features we should learn about? How do you put them together, and how can you, how do our cells use them to respond to stress, and actually how can we hijack them for other purposes? Okay, so that's the sort of the Q story. Now, there's some other interesting things about RNA modifications, codon usage, and writers that keep popping up in the literature. Okay, and well, I'll just highlight some codon data that we have on oncogenes. Okay, and I say that when I say that writers and modification and codon usage keeps popping up in the literature, it keeps popping up in cancer stories. Okay, that we're seeing. Now, one of the interesting things is if you take all the take 65 human oncogenes that are published now in a public repository, and compare them to 72 tumor suppressor genes and look at their codon usage, and that's what we're doing here. The red ones are for oncogenes, the green ones are for tumor suppressors, and really what we found is they have opposite codon usage patterns, largely opposite. 54 codons, they're directly opposing. So for oncoproteins, they're overusing this codon, whereas tumor suppressors are underusing it. So you look at the red and the greens, okay? And it's opposite. So these are your G and C ending codons. These are your A and U ending codons. So we've looked at this as a group for oncogenes. Okay. Here's another example of how to look at it. This is the heat map. Gives us a really specific view. And then this is our oncogene heat map for all 65 oncogenes. What you see is that, there's a group that is massively overusing G and C ending codons, but there's also a small group that's overusing A and U. So we think there's some regulatory features at the codon level in oncogenes and that can be backlinked to tRNA modifications, okay? Now, I'm going to highlight that Richard Gregory's lab at Harvard did some amazing work to actually link this up. Essentially was able to show that metal-1, which is a writer for N7G on specific tRNA for arginine, is massively amplified in a number of cancers. These cancers are essentially addicted to this writer and this modification, and when it's removed from the system by siRNAs and genetic means, the cancer cells die and the tumors don't form. And it's all hooked to this modification increasing tRNA stability and decoding really codons for arginine. Okay, so it's a really elegant model. You go and look at writer amplifications across cancers, you see metal-1 is amplified in a number of gliomas and liposarcomas, but there's a lot of other ones too. Okay. Again, I like to go back to the analogy with transcription factors driving gene regulatory proteins. We know they do that in different cancer models. We're starting to see it for RNA modification systems also. I just got to highlight that for metal-1, we see massive amplifications for liposarcomas and sarcomas here. Some of the enzymes that we're interested in, ALK-BH8 is massively amplified in a number of ovarian tumors. QTRT1, which you've heard about, are in pancreatic, ovarian, and a number of uterine tumors. So we're seeing this playing out in the literature as being really important for tumor growth programs. And we think it has to do with regulating the translation, increasing the translation of oncoproteins. Okay. This is a general model that's going that we built and others have also built. This is from a review that Ph.D. and I just recently put out. But the general idea is that translational dysfunction and really writer amplification or even tRNA amplification can drive translational programs that are promoting growth. Okay, we highlight this because we think these can be targeted actually as new generation therapies for cancer treatment. Okay. So with that, I know I'm probably near the end, but I want to just thank everyone for their time, and I'm happy to answer some questions.
I have a fundamental question, which is back at the beginning when you were showing your how-to. Did you isolate tRNA and then examine the modification so you separated?
Yeah. So there's two different ways to do it. You can, but you're isolating small RNA. Okay. So what we end up doing is when you isolate small RNA, it's about 95% tRNA. And so there's a QC step built in there. We use Agilent bioanalyzer chips, the small RNA analysis chips, and we do that as a QC check to see what we have in there. You get your characteristic peaks around tRNA. You also get a couple other peaks that are in there. We're saying tRNA, but it's really small RNA. We try and make the point from a biological perspective. I was talking about this morning. We want to make it easy. So what we're doing is we're picking modifications that are only found on tRNA to really do mechanistic work. So the Queuosine and the MCM5U are only found on tRNA. That's a good question. So RNA separation is always a complicated part because there's so much RNA in the cell that you got to separate the signal from the noise, and doing that is really difficult for messenger RNA. You probably know. So yeah, because the signals are tRNA is nice because the signal's high. Every single tRNA is modified multiple times. And we see about I'll go back, but I think there's like a 5 log difference in levels. There's some modifications that are found in every tRNA. There's some modifications that are only found in a couple of tRNAs. We think it's the tRNA, the modifications that are only found in a couple tRNAs that are going to be the real regulatory ones because they're different. They're regulatable.
be an exceedingly obvious question, but is codon bias defined relative to just the gene itself or to the transcriptomes?
No, that's a great question. Because codon bias, if you look at the literature, it's all over the place. And everyone defines it a little bit differently. We define it as bias relative to the genome average. Okay, and we're using a very specific form of codon bias. It's we call it isoacceptor codon bias. So this is really, is the codon bias relative to the other synonymous codons that are possible. So for sometimes that's 2, other codons, sometimes it's 6. But there's lots of measures for codon bias, and it can get confusing. It's a good question.
In your presentation, you discussed the top 50 honest genes. Can you comment on the ontological aspect of those genes, either mitochondrial or the metabolic?
Yeah, so it's a great question. So something to know about mitochondria. So they're represented in the oddest genes. Okay, so the mitochondria is weird. And I didn't know this until I had a student actually do codon analytics on the mitochondria. So all but one of the mitochondrial genes is on one strand, okay? And then there's another on the other strand. So the mitochondrial genes primarily use the U and A ending codons, okay? So they're represented in our list. Histone proteins also, okay? For the other ones, we see some transcription factors that we can link to inflammation and ROS responses as being there. We see some metabolic, but from an ontology perspective, I'd love it to be stronger. We're still thinking about it, and there's a variety, I'll say, but it's a good question.
So most of the application of the modification, you say you have empty side.
Yes.
So how do you map the modification to a specific position in the tRNA?
Yeah, so this is this is where I lean on my collaborator Ph.D. and his expertise. We have done oligonucleotide based approaches for specific modifications. And you know, the problem with RNA modifications is sequencing them is amazingly difficult. Okay, they have a number of because of their diversity and their bulk. A lot of them will promote either errors during reverse transcription or stops. Okay, so the other way that we've done is AQRNA-seq is a specific method for sequencing RNA. Pete's lab developed it, and essentially you can use that to find the stops, which you can then back map and say, well, there's likely a modification there. Other groups have also been using essentially transcription errors as a readout for modification, but it doesn't give the chemical specificity that I'm sure you would want, and I would want that too.
So all of RNA modification, they are dynamic, right? So,
It depends on how you define dynamic.
okay. So
Yeah.
in your tRNA modification, do you see for the same position, some of RNA are modified, some of them are not modified?
Yeah. So is there a yeah? We think we do. Okay. And we the way we're analyzing our RNA modifications, we're doing this, basically we're just doing a readout, right? Is the modification higher or lower? What it can mean is that you have an existing RNA that's under-modified and it's modified, right? If it goes up, right? You could have more of the RNA made as a response so that it's modified. So those are two ways for the modification to go up. And there's similar analogies for it going down. So dynamic can mean an on-off, like a write or eraser. It can be a synthesis or degradation as the other way to do it.
How conserved is codon bias?
Oh, that's a great question. Not as well as you would expect. So we've done that analysis. I got a paper manuscript that's floating around. So codon bias is more extreme in humans than it is mice. Okay. The codon bias in mice is there, but we've done for the corresponding oncogenes, and you see it, but it's not as extreme. Okay, so it might not be as big a factor in the translation of those genes. Okay, if you compare the most biased genes in humans, the most biased genes in mice, there's some overlap, but I would have expected a lot more. Okay, so it's different. And that's one of the problems is when you start using all these model systems, codon usage is different. It's different in mice, it's different in humans, it's different in rats, and these are all pretty well connected evolutionarily. When you start, so we compare budding yeast to humans, it's pretty different. Bacteria. Along these lines, when you start thinking about codon usage for different types of cancers, okay? We've another paper that we're working on is we've actually downloaded, I think, 10,000 copy number variation data sets from the tumor TCGA, okay? And compared codon usage based on copy number variation, which is really the classic way, you look at codon usage. I mean, just for the audience, when we do if we look at a normal cell, the diploid is 2. It's a perfect 2, okay? When you start looking at cancer cells, the average ploidy is like 3.8, okay? And you have like 300 copies of one gene and no copies of another gene. So we've taken all these data sets and we've asked the basic question: is codon usage significantly different in any of these cancers? And the question, and it's yes. We see it, we're seeing it, and we're still sifting through the data, but the prediction is some cancers are going to be using RNA modifications and codon bias to drive translational programs. And so the Meta-1 M7G story that came out of Richard Gregory's lab is a great example. You have an addiction, and we're going to see this, I think, in a couple different scenarios. So. Well,
tumor severity.
Yeah. We wish it was going to be. We wish it was going to be sooner rather than later, but it's going to be a while. There's the technology is we've built the tools, and now we're trying to use them more efficiently and actually get resources to do it. And it's that last part, which is always the hard part. But I mean, tRNA modifications have already been linked to health outcomes. There's a lot of weird mitochondrial diseases. So if you ever heard of MELAS, MERF, these are tRNA modification defects, among other things. So there's a lot of, I think, neuropathies and things of that nature. So the cancers are going to be a model where you have too much of a modification driving translation programs. There's a lot of intellectual disabilities, and then neuropathies and things of that nature, mitochondrial diseases, where you have less of a modification. And really what you're seeing is, those sort of cell dying phenotypes.
I guess on the alpha PHA deficiency, does that have any on?
In humans?
Yeah.
So yeah, it does, and it's actually, it corresponds to an intellectual disability in a population in, I think it's Kuwait, which is likely due to, we think, is ROS detoxification. But we also see there's some work showing that in bladder cancers, amplification of alpha PHA is driving tumor growth. But it's starting to come out. It's starting to come out in a lot of different systems. So from our estimates, there's around 50 different writers to think about with regards to RNA modifications. A lot of amazing work has been doing on the M6A. I mean, you're seeing that for different types of stem cell and developmental programs. There's evidence for different cancer programs for M5C. So we're starting to see it in the literature. Good question, thank you.
So you showed us in the acetaminophen data that there's subset of single protein, so the proteins go up, and that's dependent on the writer. I wonder if we had a different stress, you'd see a different subset, and that would be dependent on the same diet.
Yeah, I think you would. Because this, this translational output that we're looking at is really going to be very well dependent on mRNA levels. So I think you can, and in the yeast, there's ways to rescue modification defects. So you can massively overexpress at the transcript level, but you can also overexpress tRNAs that are not modified. So you can drive it that way. But I would predict that as the case there, what you say that if you use a different stress, you'd see a different subset. We've done it for naphthalene in the lung. Right? I think we see a different subset. I can go back to it. It's published, but yeah. So in addition to this, the stress, I think the tissue is also going to be a variable. Right? Thank you. Appreciate your time.